Good morning, everyone. My name is Tanmay Bachhav. I'm a PhD scholar from North Maharashtra University, Jalgaon. Today, I will be presenting my work on the copper nanoparticles in aquatic system, its biosynthesis, toxicity, and ecological concerns. So, this presentation covers our recent attempts at green synthesis and our primary observations regarding its impact on the aquatic system. So, first of all, what are the nanomaterials or the copper nanomaterial? Nanomaterials are the materials which at least have one dimension between 1 to 100 nanometers. So, the copper nanoparticles are currently being produced at the massive scale for their use in antimicrobial coatings, sensors, and agricultures. However, this industrial boom had a downside, the inevitable release of these particles into our water bodies, because the final fate of that nanomaterials into the aquatic system. So, our research addresses a critical gap understanding how these engineered particles affect the fundamental microorganism in aquatic system that form the base of the aquatic food web. So, first of all, why to study copper nanoparticles toxicity to aquatic life? So, as we know, some of the primary producers like the phytoplanktons and the microalgae are the keystone species uh, of the food webs. So, when the copper nanoparticles are widely used in the industrial like antimicrobials or they as a catalyst, they released into the water bodies, and these high release of copper nanoparticles in the water bodies causes the toxicity to algae, to the fishes, etc. So understanding these effect of these toxic effect of copper nanoparticles in unicellular eukaryotes informs the ecosystem level risk. So these are some of the key forms of copper nanoparticles. For example, elemental copper, copper oxide nanoparticles, or the cupric ions. So when the aggregates, they cause the particle specific effect to the aquatic animals. For example, physical blockage. And then the dissolution of these copper ions causes the ionic toxicity to the aquatic system, and the oxidized copper particles causes the reactive oxygen species generation, leads to the oxidative damage and forms the uh, DNA damage. These are some of the literature review related to the Paramecium chlorella symbiotic system, which is live in the aquatic system. So, uh, when copper nanoparticles are treated to the that symbiont, the growth inhibition noted at the 0.5 to 5 milligram per liter, and the damage observed because of the dual stress pathway, because the paramecium and chlorella, they both get inhibited and they are both sensitive to that copper nanoparticles. Then the second study is on the chlorella vulgaris. We show the EC50, which is the effective concentration is around 1.2 to 1.6 milligram per liter, and the uh, uh, okay, so some of the dissolved organic matter, for example, humic acid, which also reduces the toxicity of that copper nanoparticles. When the humic substances bind with that copper nanoparticles, they aggregate and reduce the toxicity of that, or the reduce the accumulation of that copper nanoparticles into the aquatic fauna. So, copper nanoparticle also. Uh, toxic to that aquatic animals, as well as also reduces the toxicity with with binding with the some of the unique materials. So first of all, our approach is the green synthesis of that copper nanoparticles, because the chemical synthesis method produces various toxic byproducts. So we chose a green synthesis route for this study. Unlike chemical methods that use toxic reducing agents, we utilize plant extract. So this approach is not only eco-friendly and cost effective, but also uses natural biomolecules and the capping agents to stabilize the copper nanoparticles. Our goal was to find a local plant species that could efficiently reduce copper ions to metallic nanoparticles without hazardous byproducts. So, to find the best source, we screen several local plants, including Tridex procumbens and various jujube species. While these plants show some activity, they often resulted in unstable particles or slow reaction times. However, we achieve excellent results with the Vernonia cinerea, commonly known as the Sahadevi. The leaf extract of Vernonia acted as a superior reducing agent, giving us a rapid and stable color change, which led us uh, to select it for the final synthesis protocol. So, after the synthesis of that nanoparticles, we go for the primary characterization through UV visible spectrophotometry. So, we successfully completed the primary confirmation using UV visible spectroscopy. We observed the characteristic surface plasmon resonance, which is the SPR peak in the range of near about the 560 to 580 nanometer, which confirms the formation of copper nanoparticles. These two graph shows the UV visible spectrum of Vernonia cinerea leaves extract, and the second diagram shows the UV visible spectrum of biosynthesized copper nanoparticles. So after that, moving to the toxicity, we first tested the particles. Before moving to that, I would like to mention that we are currently working on advanced characterization techniques, including XRD and TEM, to further define the species or the precise crystalline structure and synthesize size distribution of these particles. So first of all, we move to the toxicity. So we first tested the particles on the ciliate model paramecium. Our primary observation indicate acute stress. We noted significant behavioral changes, specifically a loss of motility and spinning movements. Morphologically, the cells show deformation and blistering of that pellicle. At higher concentration, we observe cell lysis where the membrane completely ruptured. Then the we observe the Euglena gracilis. This organism is interesting because it shares characteristic of both plants and animals. Upon exposure, Euglena cells lose their typical spindle shape and become round or cyst-like. We also observe bleaching, a loss of green color, suggesting that the nanoparticles are physically damaging the chloroplast and interfering with the photosynthesis. Then the our third model involved diatoms. These are vital for aquatic oxygen production. We observe that the nanoparticles cause the deformities in the silica shells or frustules. We also noted a stress response where the diatoms begin accumulating lipid droplets inside the cell. Additionally, the particles cause the diatoms to clump together, which physically hinders their ability to float and access nutrients. So currently, our data is based on the strong microscopic observations. So we are now expanding this work to understand the molecular mechanism behind these changes. We hypothesize the reactive oxygen species generation is the main driver of this damage. We are setting up assays to measure the oxidative stress markers to confirm these hypothesis in the next phase of our research. So the ecological implication here is the potential for trophic transfer. If primary producers like diatoms accumulate these nanoparticles, they can be eaten by small crustaceans and eventually fish. This suggests that the copper nanoparticles could biomagnify up the food chain and uh, posing risk not just to algae but to the entire ecosystem. So, to conclude, we successfully identified Vernonia cinerea as a potent agent for the green synthesis of copper nanoparticles. However, our biological screening shows these nanoparticles induce several morphological and uh, uh, behavioral changes in aquatic system. As we continue to characterize the particles and uncover the toxicity mechanism, it is clear that the environmental release of these nanomaterials requires careful monitoring. So these are some of the photographs of our work with my guide. These are some of the cultures we are maintaining at our laboratory. So these are the references, and thank you.